guys, my name is Abhishek and welcome back to MBBS Treasure Channel. Today we are going to read the topic that is disorder of immunological reactivity. In my previous video, I have already explained what is immunological reactivity, and in this topic we are going to read the disorder of immunological reactivity. As I have said, the immunological reactivity means it's a reaction of our body through the immune system protecting against the antigens by producing the antibodies or by different cellular reactions. Okay, so if there will be problem or disorder in this immunological reactivity, then we will call it as immunological insufficiency. Okay, so what is the definition of this immunological insufficiency? First, it is hereditary or it is acquired defect of the immune system that is characterized by insufficiency of organisms to carry out the reaction of humoral and cellular immunity variably. Okay, so here the definition says that it is hereditary or it can be acquired. It means it is congenital before birth or it is acquired during the process of ontogenesis. This disorder may persist. Okay, so what this means? This defect of the immune system, there will be disorder of this immune system, or the immune system will get hampered, through which the immunological insufficiency will occur. And how does this characterize? It is characterized by the insufficiency of an organisms. It means that the organisms will not be able to carry out the normal reactions of the immunological reactivity, that is humoral reaction and cellular reaction. As we know that humoral reaction is regulated or mediated by B lymphocytes and the cellular reaction is mediated by T lymphocytes. So what will happen if there will be disorder in this immune system? Then there will be problem lying with this immunopotent cells or the cells of the immunity. So as the problems lies with these cells, then only the disorder will lead to this immunological insufficiency. Okay, now comes the classification of immunological insufficiency. How this immunological disorder is being classified. The classification says here is that it can be primary hereditary like this. It can be primary hereditary or it can be acquired. It means it can be secondary. Primary means the thing that will occur first, and the secondary means it may occur after the second thing occurs. Okay, primary means it will be hereditary, it may be congenital, it may be present before the birth, and how it occurs if the defects, innate defects of the immune system. And secondary will occur if the defects are acquired during the process of ontogenesis or the development of an organism. If the defects are acquired, then we will call it as secondary or acquired type of immunoinsufficiency. Okay, now comes the reason why this primary hereditary occurs, or this hereditary immunological insufficiency occurs. The reason here is that there will be change in DNA sequence during the process of cell division. Listen, if any problem or disorder lies in during the process of cell division, it means during the process of gametogenesis or the formation or the development of fetus embryo, at that specific time this disorder will occur. And secondary means after the birth of the child. This change in DNA sequence is nothing but mutation. So the mutation will be the primary causing factor of the hereditary immunological insufficiency. So it can be gene mutation, it can be chromosomal mutation, and thirdly there lies one more thing, that is prenatal infections. So these three terms will cause this primary or hereditary immunological insufficiency. And according to gene mutation, it is further divided into two types, that it may be like sex-linked mutation or it can be autosomal recessive mutations. And chromosomal mutations like translocation, inversion, deletion and duplication. This genes mutation, I will explain it separately in my next slide. So now let's come to the chromosomal mutation. Listen, chromosomal mutation, what do you mean by this? The mutation that will be occurring in chromosomes during the process of gametogenesis. What happens? The cell division takes place, there will be replications of the DNA, and also the chromosomes that are present in DNA will also get replicated. Similarly, during that procedure only, this mutation will occur, and if we will go more subclassical division, then the gene mutation will occur. Okay, this chromosomal mutation is divided according to the errors of this crossing over and errors of this replication. So as there is a chromosomes, during the process of cell division, it will what? It will get replicated and it will get crossed over with one another during forming of pairs. Okay, so what will happen during this procedure? The chromosome will get mutated. So how the mutation is divided in chromosomes? It is divided during crossing over and replication. Let's say for an example, this is a segment of DNA and it consists of parts like this A, B, C, D, E. First one is deletion. Let's say deletion. What do you mean by deletion? Loss of a chromosomal segment. If this is a segment, it will get lost during the process of replication. What do you mean by this? After the process of replication, we will not find this B component, A, C, D and E. So this process is known as deletion. Next, what happens? If there will be duplications of this special gene B, then what will happen? Then the B segment will come twice. Okay, this B segment will come twice because duplications means repetition of a segment. Okay, and this inversion and translocation, this we read about. Now comes what is translocation and inversion. Let's say this is one more segment. Okay, if the process of inversion will takes place, what will happen? This will get reversed 180 degree. It means it will be E, D, C and B and A. Okay, so there will be complete reversal of the segment of DNA, that is known as inversion or reverse of a segment by 180 degree. If there will be translocation, what do you mean by this? The movement of a segment from one chromosomes to another. Let's say this, let's see this. Here is one segment and here is another segment. Okay, here is another segment, A, B, C, D and it is E, F, G, H. So what will happen during the process of translocation? This let's say this D part of the first chromosome will get transported. It means trans means transported in location. There will be changing of location from the initial chromosome to another chromosomes. So the D part will get transported to another chromosomes, resulting in, this will result in E, F, G, H and D, and here what will happen? It will be only A, B and C. Okay, so there will be adding up of one more gene of the chromosomes to one more chromosomes, and there will be losing of one gene from one chromosomes. So D can be placed here, D can be placed here. So wherever it may change its locations. So this is nothing but as translocation. So as we know the chromosomal mutations, the translocation, inversion, deletion and duplications, the translocations takes place at some kind of leukemia, and inversion will take place like in hemophilia A. There will be the inversion of the gene in the blood clotting factor 8, that is known as hemophilia A. The deletion can take place at the Credichet syndrome. The example of the deletion is Credichet syndrome, in which there will be loss of or the deletion of a chromosome number 5. Okay, so there will be Credichet syndrome, and finally the duplication. Duplications mostly occurs at the tumors, the cells that will be continuously dividing and producing more number of replicated cells. Okay, during the process of tumors, the duplications will take place. Okay, so next comes here is the prenatal infection. What do you mean by prenatal infections? The infection that will cause the hereditary changes or the hereditary immunological insufficiency before birth. During the period of uterine growth, if the baby will get infected through some kind of infections, that is like the torch infections. In a short abbreviations, we can call it as torch. It means TO means Toxoplasmosis gondii, R means rubella virus, C means cytomegalovirus, and H means herpes simplex virus. So these are the infectious causing agents that will cause the infections in the birth, and they can pass through the placenta from the mother to the child. Through that process, the developing embryo or the fetus will get infected, and that will cause this disorder in the immune system. It may affect the B cells, T lymphocyte cells, through which after the birth the baby can never show the immunological reactions to the antigens. Next comes is the secondary or acquired. As I discussed, the defects are acquired during the process of development, and the main reasons are the mutagens. Mutagens are nothing but these are the factors that will cause the mutations. These are the external factors. It can be exogenous like the ionizing radiations, it can be chemical and biological factors, and in case of endogenous, in our process of development we find aging and intoxication with some of our biochemicals that are present in our body, that will tend to cause the defect in the immunological cells, and that is known as secondary or acquired immunological insufficiency. Okay, so this is the basic classification of immunological insufficiency. Next, we are going to read about this gene mutation, and more specifically this sex-linked mutation and autosomal recessive mutations, or disorder relating to this